Hey, my name is Keshav, and today I'm going to be talking about favorable mutations of polymerase orthologs and how we can introduce them to human DNA polymerase. So today I will be talking about a couple of mutations and their applications and wrap this all up with talk about ethics. DNA polymerase is a vital protein in the nucleus of every cell, and it acts by adding nucleotides to the split strands of DNA during replication. Four nucleotides that it adds are adenine, guanine, cytosine, and thymine. The problem with DNA polymerase is it makes about one error per every billion nucleotides it adds. But this problem completely compounds when you consider the sheer number of genes that are present in the human body. DNA polymerase can also halt and stop at any given moment, while in replication if it encounters a strand that is too difficult. This could lead to mutations, which are very dangerous and exhibit themselves in horrible ways sometimes, risking diseases like sickle cell or cancer. The solution I've come up with are other types of polymerases, because when you look at other organisms, they have differently naturally occurring types of DNA polymerase. Some of them have differences with different characteristics that can get around these issues. None of these have ever been considered for human in use, obviously, since they're made by completely different organisms. But researchers have been able to produce polymerases in the laboratory and generate more variants, which means that it could possibly be within our scope of potential to integrate these into humans. T4 DNA polymerase and L421 M mutation is a particularly interesting variation of this. T4 DNA polymerase comes from the T4 bacteriophage, a common virus that infects E. coli bacteria. It serves a pretty important function in DNA replication and repair in the bacteriophage, since the bacteriophage mainly hijacks other bacteria and replicates that way. It also possesses a really robust and proofreading activity, which lets it remove incorrect sequences very rapidly, and it's used a lot in molecular biology. A specific mutation discovered by uh, a certain doctor a couple years ago, L412M mutation, was a pretty big discovery as it was found to enhance polymerase plasticity by copying longer strands of DNA without falling off the strand. As you can see here, T4 DNA polymerase is shown, and this is this particular gene. Since this gene has increased processivity, it means that it can easily process and with less delay can scan the genome to make sure that the protein functions properly, which reduces the error rate. This could be translated to human medicine and biotechnology, as these are really good conditions under stressful uh, situations. It could be used to develop highly processive DNA polymerases for our bodies or even just for lab use. And this would be pretty useful in therapeutic or genetic disease treatment areas. T4 DNA polymerase can also be used in CRISPR. For those of you who don't know what CRISPR is, it's a gene editing sequence that is used to edit DNA using a protein called Cas9. It cuts the DNA sequence and replaces it with plasmid, which has different DNA. As you can see in this image right here, this is the left side represents the traditional CRISPR process, while the right side uses the T4 DNA polymerases attached to the Cas9, which makes sure that if there are on-target mutations or off-target mutations, which are unintended edits on the site of the target, the T4 DNA polymerase can repair and uh, fix that exact area to make sure there are less mutations. Now, my last section will talk about how different mutations could be introduced to human polymerases. Obviously, if we're using this using traditional methods like CRISPR or Cas9, we can always delete different parts of it or go completely off. So we should plan this and make sure we fully map out the sequence of the genome that we want to edit this in. For example, if you wanted to reproduce the L412M mutation in human body cells, the final target, you would have to produce a map showing the difference between the human and T4 DNA polymerase and how you would intend to edit the human version in order to produce the same results as the L412M presents in T4 DNA bacteriophages. Ethical issues also exist. As we know, somatic editing is a pretty new concept. It's been around for a while, but even though they're not inherited and they're not germline changes, the irreversibility of some of these changes makes it very risky. Also, patients may not know what they're consenting to. So, as we discover more information about this, we should make sure to keep the public updated and make sure all patients for this process know what they're getting into. Another ethical question is if there's the process of somatic editing could go into physical or mental enhancement traits, which on its own may not seem as harmful as you may think, but this could lead to eugenics and different gene editing technology used by the government. So, to make sure this doesn't happen, we should, as researchers in the scientific community, have informed consent, equitable access, and wise regulation in process for these uh, editing processes so that the general public knows what's going on and we don't have imposed uh, editing on us. This is a larger problem with gene editing in general. So in conclusion, L412M, it's a specific DNA polymerase mutation that could have edited gene editing efficiency. This shows that there are processes in polymerases like T4 DNA which can help us understand and even better our own. Um, Cas Plus is also showing that we can work with these polymerases by themselves in order to generate better systems. However, with these powerful changes, we need to make sure that we're not destroying our own world by creating a social problem or even just hurting other people unintentionally. Thank you for listening to my presentation.